Welcome back to the Vitals Podcast. I'm your host, Chauncey, back again to digest and bring you the latest in medical news today. It is Monday, July 15th, 2024, and I have more discoveries for you today. As always, don't forget to follow the podcast. Let's get to 100 followers. Share it with your friends to keep everyone you know in the loop. Another H5N1 case has popped up in Colorado, and as with other cases, the person reported eye symptoms only, received oseltamivir treatment, and has recovered. The CDC has been watching influenza surveillance systems closely, especially in affected states. And there has been no sign of unusual influenza activity in people. The CDC keeps the health risk assessment at low, but continues to emphasize the importance of using PPE. People with close or prolonged unprotected exposures to infected birds, other animals, including livestock, or environments contaminated by infected birds or other animals are at greater risk of infection. Our first story comes from Dr. Stephen A. Rosenberg and other NIH researchers at the NCI's Center for Cancer Research, or CCR, who are studying the effectiveness of immunotherapy against solid tumors. One form of cellular immunotherapy, chimeric antigen receptor, or CAR T-cell therapy, has already shown to be effective against some blood cancers, and another, called tumor-infiltrating lymphocyte, or TIL therapy, has proven to be effective against metastatic melanoma. However, to date, a cellular therapy effective against any other solid cancers has been elusive, according to Dr. Rosenberg, who co-led the study with Dr. Maria Parkhurst of CCR's surgery branch. The new approach overcomes two challenges in cellular immunotherapy: how to produce large numbers of special T cells that can recognize cancer cells specifically, and how to boost the ability of these particular T cells to multiply once they've been returned to the patient. For each patient in the study, Dr. Rosenberg and his colleagues collected white blood cells called lymphocytes in the patient's tumors. They then used specific techniques to identify and isolate receptors on those lymphocytes, called T-cell receptors, that recognize specific changes in each patient's tumor compared to their healthy cells. After reading and replicating the DNA that makes those receptors, they used a lab-made package called a retrovirus to insert the genes for the receptor into normal lymphocytes collected from each patient's circulating blood. The genetically modified lymphocytes were then multiplied into hundreds of millions in the laboratory and infused back into the patients, where they expressed the tumor-specific T-cell receptors and continued to multiply. In short, they took natural T-cells from the human body, found what T-cells targeted tumors, replicated what made them target tumors, and inserted those special T-cells back into the body. Dr. Rosenberg explains, quote, by taking the natural T-cells receptors that are present in a very small number of cells and putting them into normal lymphocytes, for which we have enormous numbers, 1 million in every thimbleful of blood, we can generate as many cancer-fighting cells as we want, end quote. As part of a larger phase 2 trial, 7 patients with metastatic colon cancer were treated with the experimental personalized cellular immunotherapy. All 7 received several doses of the immunotherapy drug pembrolizumab, or Keytruda as it's marketed, before the cell therapy and another immunotherapy drug called IL-2 afterward. 3 patients had substantial shrinkage of metastatic tumors in the liver, lung, and lymph nodes that lasted for 4 to 7 months. The median time to disease progression in comparison was 4.6 months. Dr. Rosenberg noted that 2 of the 3 patients who responded to the treatment had received those retrovirus packages with the DNA from cytotoxic T cells, which are special T cells primarily responsible for killing disease cells, like cancer cells. Dr. Rosenberg said his research team is exploring how to put the T-cell receptors into subtypes of normal lymphocytes to improve their reactivity. Colon cancer is just one of many solid tumors the researchers are studying. The trial is still ongoing and includes patients with different types of solid cancers. Dr. Rosenberg says, quote, it's just the very beginning of converting normal lymphocytes into cells capable of treating the common solid cancers. What the study shows is that it's possible. Once you know it's possible, you work to improve it, end quote. Our second story comes from Dr. Richard Krauzlis and other NIH researchers at the NIH's National Eye Institute, who have discovered a new circuit in brains that helps us recognize faces. The newly discovered circuit first engages an evolutionarily ancient part of the brain called the superior colliculus, which can then trigger the eyes and head to turn for a better look. This better view enables different brain areas in the temporal cortex to engage in more complex facial recognition. In adult primates, the brain develops specialized regions of the temporal cortex called face patches that provide the ability to recognize and distinguish individuals by their facial features. However, facial recognition depends on fine details provided by the eye's high acuity central vision. For us to recognize a face, we first have to look directly at it. When babies are born, they lack the high acuity vision needed to see the fine details of faces, and the face-specific areas of the cortex don't develop until later. Even so, babies generally orient and look at faces very early in life, suggesting that another process is in play. Krauzlis and colleagues hypothesized that the superior colliculus, known to detect objects, could provide the missing link. Part of the midbrain, it tells the rest of the brain if something is present somewhere. Not what the object is, but simply that it is there. It functions extremely quickly and connects directly to the brain's motor parts, directing eye movements towards objects of interest or flinching out of the way of an object in the peripheral vision. To test whether the superior colliculus might aid in face detection specifically, co-first authors Dr. Gongchen Yu and Dr. Leo Katz assembled a collection of images including faces, biological non-face objects like hands and arms, and other items like fruit or human-made objects. They then showed these images to adult monkeys in the monkey's peripheral vision field and recorded neuronal responses in the superior colliculus. They found that within 40 milliseconds, more than half of the neurons they measured responded more strongly to images of faces compared to other types of objects. Some additional neurons eventually displayed preferences for other types of objects, but not until 100 milliseconds after. In other words, the face-specific detection was much faster than detection of other objects and was preferred by a large proportion of the measured neurons. The researchers were also able to determine that, while the superior colliculus can receive visual information directly from the eye, its object detection process instead requires input from the early part of the visual cortex first. Since the superior colliculus also reconnects back to the visual cortex later on in processing what we see, the scientists suspect that this circuit provides a mechanism to highlight the importance of certain objects. The findings help not only explain how primates sense and recognize faces, but could also have implications for understanding conditions such as autism, where face detection and recognition are often impaired from early childhood. There's no vital question this week, so that's all for this week's episode. Thank you for tuning in. Follow the podcast or share this episode with your friends, and if you'd like your questions answered, remember to drop a response in the Q&A section of this video, and you may see it in a future episode. Quick note before you leave though, I want to invite you to follow me on Goodpods. If you haven't already heard of it, it's like Goodreads or Instagram for podcasts. It's new, it's social, it's different, and it's growing really fast. I'm excited to hear your feedback and questions about the show. You can find Goodpods on the web at goodpods.com, download the app, or use the link in the description. My username is @CJTaylor. Follow me and let's grow the community. Until next time, keep your vitals strong and your spirits high. This is your host, Chauncey, signing off. Take care and stay healthy. I'll see you next week.